business that they looked into breast cancer, they looked into multiple sclerosis, they looked into Alzheimer's, cystic fibrosis, how each of these have succeeded in fundraising, and they gave me some input to share with you today. Today is basically a brainstorming, and then the second half of our discussions today with my commentary is to put together the vaccine discussion forum, a grant that we proposed to Pfizer. We were happy with Mike's support to get initial green light. Now we have to put more details into it, how to build a vaccine discussion forum. And the third goal of our meeting is AEHA is going to set aside a small amount of funding until we're supported in a larger bulk to write up a supplement to Scientific American Journal. So sort of finding some of the time that should go into writing for this, and I was hoping that Jan would take the leadership on this executive summary, and with your help and everybody else, we will finish it and put a milestone for, say, 6 months with these topics and these highlights. So these three are our goal today. One is cover science. You bring us to vaccine or immune modulation for dummies. We get that level. And then the second one would be discussing the successful experience by other similar situations, particularly the one that I gained recently a lot of knowledge was Alzheimer and cystic fibrosis. And I have some good news about Alzheimer and how they're really pushing this forward with this new politics, right? And then again, finally, we will nail down the Scientific American Executive Summary or supplement for vaccine. Any comment? Would you like to change the agenda? Or?
No. I think that sounds fine. I mean, just by way of introduction, again, as I mentioned the other day, this whole idea of vaccine really began in 1995, 96. 92. 92? Our first paper was in 96. Jan was with us at Cedars on a sabbatical, and he first showed that using oxidized LDL as an antigen actually reduced atherosclerosis in the rabbit model. And this was basically LDL taken from one group of rabbits, copper oxidized, put together with an adjuvant and given as a vaccine. And right around the same time, Wolf Polinsky with Joe Whitstone published their paper in a Watanabe rabbit using MDA-modified LDL, which is really taking LDL and using MDA modification, and showed exactly virtually the same results in a totally different model, independently. This led to the idea that oxidized LDL has antigens in it that can provoke a protective immune response. And the question was, how can this be ever brought to the point where it can be a clinically testable strategy? So over the last 5 years, Jan's group and our group have been working on identifying antigens within the protein part of LDL that could provoke a protective response. And so using a very ambitious approach of designing 300 peptide sequences that span the entire ApoB100, and then choosing the ones that human blood reacts to, and then out of that choosing a few random sequences to test as vaccines, we stumbled upon at least 4 sequences, 2 in Jan's lab, 2 in our lab, that actually, when incorporated as a vaccine with alum is used clinically as an adjuvant, produced anywhere from 50% to 70% reduction in atherosclerosis in the ApoE knockout mouse, and changed the plaque phenotype with less lipid and less inflammation.
Okay, was that prevention of...
progression, yeah.
No, Were these mice that already had it?
No, this was
reduction.
actually starting at age 6 to 7 weeks.
So you prevented the development.
Development, yeah, we have not yet, we have plans to actually look at later stages, but this was early stage atherosclerosis. And cholesterol levels didn't really change very much. And contemporaneously, Polinsky's group showed that phospholipid antigens are also can provoke a protective response, but the magnitude of that protection seemed much less than the ApoB-related antigens. And we have confirmed that recently in our lab using phosphorylcholine, antibody to phosphorylcholine in a passive immunization. It doesn't do as much as the active immunization with ApoB. And I think at this point maybe we should turn to Jan to actually show.
Before we enter into a systematic education, you look at PubMed, the immune component of atherosclerosis is, go back like 50-some years.
150
minimum organized. When immunology has developed. Yeah, 150 years ago, Workhouse said PC's macrophage type or white cells. And then, if you follow this literature, there's really interesting sparks. Somebody found this here and there, and somebody started with this virus-related complement deposition plaque. And if you, I need your help to get us into a basic level of education. In this process of development of knowledge of atherosclerosis and immune, where was a severe degree or a serious degree of immune complement or immune component of atherosclerosis has evolved? I mean, we're obviously going back to 70s or 60s. Can you tell us a little bit about it? Because the reason I'm bringing this up is there can be some learning.
Yeah, historically, I think all components of the immune system exist in the atherosclerotic lesion. T cells, macrophages, dendritic cells, immunoglobulins, complement, antigen, antibody. These can all be demonstrated in the atherosclerotic lesion.
Mast cells.
Mast cells, exactly. All the effector cells of the immune system. That's anatomic evidence, right?
There are very few B cells.
There are very few B cells,
but lots of antibody.
Yes,
antibody, absolutely.
And immunocomplement depositions.
They come from the blood.
Now, all components of the immune system are there within the atherosclerotic lesion. And then I think it was Freigang or Jan's group showed many years ago that T cells reactive to oxidized LDL could be demonstrated. I don't remember whether it was your group or Goran Hansen.
Goran's group was the first to report it.
That you could demonstrate T cells that had been sensitized to oxidized LDL within the atherosclerotic lesion. Okay, so there was clear indication that there was an immunological recognition of oxidized LDL, and you had T cells that reacted to oxidized LDL. That was shown many years ago.
It was 85. Yeah, 85. Yeah, that's the paper we also.
And then antibodies to oxidized LDL were demonstrated in circulating blood of humans and animal models. So we knew there's a human immune response to oxidized LDL. And then if you use two other antigens that have been recognized, autoantigens, one is beta-2 glycoprotein 1. If you immunize mice against that, rabbits or mice against that, you get acceleration of atherosclerosis. And if you induce tolerance to that antigen by mucosal delivery of the antigen, you get less atherosclerosis.
Yes, we're going to go to the antigen levels because you probably have some other potential candidates that others think. So this series of observation, they weren't organized, of course, observation. There was somebody
from different
this guy in, I don't know, one of the Scandinavian car, he's obsessed with mast cells, and he only sees
Patrick Collins.
I love it. He just published a new one. And then somebody, I think Goran Hansen was deep into T cell interactions and trying. Now, At one, see, so this is like 30 years of observations coming through this. At one point, if you look at literature, you see contradictory reports about antibodies. That immunization increases atherosclerosis versus immunization decreases atherosclerosis.
Well, the oxidized LDL immunization has always reduced atherosclerosis. It has never been shown to accelerate. It has always shown reduction.
When you look on association studies with antibodies in humans, then you come up
with disparity. But active immunization has always reduced that in every single one.
So just retrospective or correlation studies disparities that sometimes excess antibody was associated with increased atherosclerosis.
Exactly.
Sometimes excess antibody was associated with decrease, which that's an interesting question.
But the actual immunization experiments consistently from all laboratories, there are basically, 3 labs that have focused on our lab, jointly, UC San Diego, and the group in Israel, Dora Haratz and his group, have been looking at immunization.
Immunization against oxidized LDL?
Oxidized LDL, some have looked at beta-glucoprotein and Hitchcock protein. Those two antigens, immunization always increases atherosclerosis. Okay? So beta-2-glycoprotein and Hitchcock protein are pro-atherogenic autoantigens. Oxidized LDL is different.
Because you read this, this is very good. So are we limited to these 3 now?
These are the known.
The 3 known, but Hitchcock protein is not necessarily specific to atherosclerosis, because we have Hitchcock protein antibody and immunocomplex in rheumatoid arthritis. Right. We have something in MS. They're talking about in MS. So it's I think George Week.
He's been a big proponent.
Yeah, he brought this up as the unified hypothesis around all autoantibody diseases. But beta-glucoprotein, is there any other diseases that share this?
Yes, it's in the antiphospholipid syndrome, like lupus.
Okay, lupus.
Yeah, it's an autoantigen there.
And to your knowledge, there is no other diseases that has autoantibody against oxidized LDL that would...
It's not really been studied.
Not
it wouldn't surprise me if you would come up with a number of other diseases where you have chronic inflammation in the tissue, for example, rheumatoid arthritis, that you would have lipid oxidation there, you would have generation of antibodies, which presumably might influence that disease too.
And
Alzheimer. So I think for us to just come to a ground that we can understand.
Put the topic into perspective. I mean, immunologists now recognize that there's an innate immune system and there's the adaptive immune system, and they are linked. An innate immune system is when you get a foreign invasion by a bacteria, there's a group of immune system that reacts immediately against that, not having any memory of it, but acts immediately. There's a blunt response.
There are soldiers on the border.
Exactly. They are ready to go.
Yeah.
And that has that reaction is mediated by toll-like receptors, which eventually leads to NF-kappa B mediated inflammatory response. That's a blunt response due to pattern recognition receptor. One component of that is we all have naturally existing antibodies against antigens we have not been previously exposed to.
These are general bullet that should
Those are called, innate antibodies, like they're IgM.
Genetic?
IgM, yeah, we all have them. They've been recognized.
Genetically determined from, you can trace it back to
exactly.
to DNA.
And it turns out those antibodies recognize phospholipid antigens that are exposed during cell death.
I see.
That are present in the cell wall of pneumococcus bacteria.
I see.
And it turns out some of the oxidized LDL antigens also, these antibodies also react with the phospholipid antigens within the oxidized LDL. And they're pre-existing. We have them.
This is nothing you've developed after exposure.
These are innate.
This probably came from our evolution.
Yeah, because it was probably designed to clear out apoptotic cells.
They're causing the glimpses. They're causing the glimpses.
Right.
Okay.
Because that's the innate.
Because I think that I'm getting this one is, this innate pathway is a very labor-consuming pathway, comparing to the humoral, which is more intelligent, because you have...
This is blood.
you have to fight with it.
Right.
So I think over time, Mother Nature empowered us with this part of
This is our first line of defense.
part of this empowered us with some of these, the kind of guns that you just shoot and it keeps going on its own, automatic. So you have that general IgG as a part of it to clean your apoptotic IgM. So it is only IgM.
Right, the natural antibody is an IgM.
Only IgM. To clean your apoptotic bodies, because that happens all the time, and your body exposes all those debris, and cleaning them by hand in that macrophage system is a very labor-consuming. So probably...
Now, having said that, so this is the innate immune system. Question is, what is its role in atherosclerosis? It turns out most of the innate immune system actually plays a pro-atherogenic role because of NF-kappa B mediated inflammatory responses. And we have shown recently the toll-like receptor and its downstream adapter molecules are highly pro-atherogenic. If you knock them out, you get a large reduction in atherosclerosis.
Teleologically, you would want to expect that this is anti-inflammatory.
No, this is a product, because inflammation is a response to an offense. And inflammation, the idea is to clear it up.
Right.
So it is actually defense against infection. Right, but when it actually goes awry and actually produces inflammation, which then actually accelerates atherosclerosis.
Well, if you said this natural IgM...
Right, that's the only exception.
Oh, okay.
So the cellular response in the
that's
immune
inflammatory.
system
That
is inflammatory.
It's fire. It's war.
The IgM, natural IgM, is partially protective.
Okay.
So that component of the immune system is partially protective. So if you immunize with pneumococcal antigen, which contains the phosphorylcholine, you get increase in this IgM, which also cross-reacts with oxidized LDL phospholipid.
Okay.
And that is partially protective.
When you say protective, means
Reduces atherosclerosis.
because it cleans up the oxidized LDL from the
It
system?
actually prevents the uptake of oxidized LDL by the macrophage.
We're not really sure about how the mechanism...
The exact mechanism is not known.
But at least if you measure circulating oxidized LDL, you would expect that goes down or not change. If it doesn't
It's
change,
much more complicated than that. I don't think it's as simple as...
or even oxidized LDL in the plaque.
Now, the only thing that's been shown that is, the antibody is bind to phospholipid within the oxidized LDL and reduce its uptake by the macrophage. That may be the mechanism.
Okay. So this is the
innate. Then the adaptive immune response is when your body recognizes an antigen and it's presented by dendritic cells and macrophages to the T cells.
T cells.
Through various receptors, and they develop then a specific response, and that specific response can be cell-mediated or antibody. Okay, and there's evidence from all the work that this adaptive response to oxidized LDL antigens is protective.
Again, back to protective means reducing oxidized LDL or reducing plaque.
Reducing
We don't know what happens with that.
mechanism we don't know. Is it antibody? Is it cytokine dependent? Is it TH1,
Because if the
TH2?
whole process of immunization lead to reducing the total burden of cytokines or inflammation in general, not specifically the number of oxidized LDL particle, then that's one thing. But if it leads to reducing significantly the level of oxidized LDL in circulation,
No,
and
it's not as simple as that, as you will
It's
see
not.
from the data. And the only thing we can say is spleen plays a very important role.
So
Because if you remove the spleen and when you immunize, you have no protection. And conversely, if you immunize and then take the splenocytes out,
Then the plaque
With them
will
to unimmunized mice, it conveys that protection.
So
Now the immune system is involved, but exactly the mediators.
what's in the spleen? There are
Parasites, there's all kinds of things in spleen.
I think this is, a very important point, because we've been focused very much that
On
whatever
antibodies.
goes on in atherosclerosis
Exactly.
goes on in the vascular wall.
Yeah.
Right, well, that's true.
But it may very well be that much of the important things
It's happening
goes
in the
on
spleen.
somewhere else, in the spleen, or in lymph nodes,
Yeah.
somewhere, exactly like it's liver that really determines the lipid accumulation in the vessel wall. It may be the spleen that
Determines the
determines
immune,
inflammation in the vascular wall.
Right, but what we all know that the phenomenon is localized in certain areas in the vessel wall.
But it may be influenced by what's being produced elsewhere.
Well, definitely, it is a circulating, but what happens is there has to be a local pathology.
Local factors and dynamic shift, stress and so forth.
But beyond that, there has to be an additional component to the immune system, linked to the immune system, where if either increased residence time or changing the the pattern of oxidized LDL once they get in touch with matrix and so on, which would trigger the immune system. We're going to learn this. I think we're going.
But this is so that the B cell response to oxidized LDL occur outside the vessel wall,
Yeah.
and the T cell response to oxidized
Inside.
LDL occur within the vessel wall. And then the immunoglobulin produced outside the vessel wall to oxidized LDL secondarily not go into the vessel wall and influence the process.
So, for example, hypothetically,
I wish we knew exactly, it would be easy for us.
hypothetically, if you think that's what it is, but we don't know for sure. Do we know that the T cells, when they leave, or APC cells, when they leave a plaque, they leave through lymphatic nodes?
There's actually both pathways. For macrophages, can exit the plaque to belong to the lymph nodes, or they can exit in a reverse direction into the lumen.
Do we know that?
Absolutely. There's a paper in PNA is just published last week.
It's
absolutely
this paper you're talking about, the immigration paper.
Yes, it's from Ed Fisher's group.
Yeah, but this paper doesn't tell you exactly what.
It shows evidence that both pathways are active.
This paper was an in vitro paper.
No. This is using a vessel wall model, actually.
Model, but it's not a. It doesn't have.
No. This is actual FOE knockout mice in vivo. It's in vivo.
And they show that macrophages can leave the vessel wall into the lumen again.
Now, this is what the concept is, that once macrophages go into the vessel wall, if they convert into dendritic cells, they have a good chance of exiting through the
lymphatic nodes.
Yeah.
But if you have phospholipids generated by oxidized LDL, they become immobilized
Yeah.
and they cannot exit.
And they fixed.
They get fixed. And that's so one of the hypotheses that retention of macrophage by pro-atherogenic lipids may be one of the reasons you have continued inflammatory cell accumulation. And they have evidence to show that dendritic cells can leave through the lymph node and also through the luminal side. They actually showed the data.
This is interesting. There is a paper, very beautiful
That
paper.
is a chemokine that attract them back into the
But they don't know what causes the migration.
Well, they say path ligand pathway.
That's one of the things.
That's what they say.
The interesting thing is that there is a trafficking.
There's the trafficking, but if you look at the paper by Guyton, the famous, not the famous, his son in North Carolina, like 12 years ago, 15 years ago, where he studied these rabbits with electron microscopy, and I think he was the first who showed that this macrophage
It was Garrity.
No, Garrity was before. That was your
Yeah, but you could not see, you saw a macrophage.
Yeah, but coming out of
endothelial cell, and you cannot see what way
Which way direction it's going.
So it has never been shown that macrophages, in fact, or foam cell can leave the lesion to the lumen.
Yeah, except this paper is absolutely
It's
beautiful.
dendritic cells, it's not foam cells.
Well, no, it is actually macrophages, modified macrophages. Actually. It's
But the same study Daniel Stein did with XY chromosome study, do you remember? The PCR study, where he injected from female to male and traced his study says 6,000 cells traffic. He can't say which way they go, but 6,000 cells traffic a day was the average that they found, and they just did PCR. I think the PCR approach is much better than what they did with fluorescence. This group.
Not necessarily.
Well, PCR you really measure it with.
Yeah, but the PCR also has many artifacts and many techniques.
Well, you get a piece of female injected in male, but there's no way you can find in any other cells of that. And that's what I was just getting out of. And the reason why that is, Willerson did this study with fluorescence in A4E mice, and he and AGA, and we followed up with iron particles, and every time we only see one snapshot. We can't see monitoring the movie which way they go. But don't you think the majority of them, indeed, I mean, just imagine the picture that Mayo Clinic group with Vazovazone, this network of microvessels around the plaque and deep inside the plaque that created for us. Don't you think that probably 99% of this traffic...
I don't know what the percentage is.
I mean,
the fact is they go in both directions.
Now, the reason I brought this up is if the majority of them are likely to go through that Vazovazone network and go backward, probably if you trace them through the lymphatic path, has anybody studied lymphatic path around the artery?
This paper does actually show. They isolate these.
That the lymphatic tissue has been largely ignored.
Ignored, yeah, even in myocardium.
But this is the first paper that actually shows that in the lymph nodes you can identify these cells that have migrated out of the atherosclerotic lesion.
Do you know if we open rabbit or pig or a large animal, dog, can we trace lymphatic?
Well, they're traced in ApoE knockout mice.
Do we know is the lymphatics in the lesions? Are the lymphatic vessels in
To the local lymph nodes, like in the cervical lymph nodes. That's the regional drainage.
Well, there is
lymph vessels? It's the lymph vessels.
The lymph vessels in the tissue.
In the
tissue, yeah.
But we have blood vessels in the plaque at the base of the plaque.
Right.
We know that angiogenesis, but do we know whether we have also angiogenesis of the lymphatic vessels in plaque?
Lymphangiogenesis.
Yes.
I don't know.
Well, actually, yeah, there is. I actually hired a surgeon, a Russian surgeon in my lab 2 years ago. He did all his life lymphatic anatomy. That's all he did for in his lab. And when he joined us, he was obsessed about, nobody has studied lymphatic system and his point was he didn't know about this. And the reason I brought this up is if we are able to cut it or to put something there and measure the antibody level, we can trace what you say you don't know between systemic and local degree of immune response, or either antibody or whatever we want to talk about. I think we're ready now to listen to your presentation.
Introduction. And coming back to your question when sort of the immune system came into focus, I think it started after Göran's investigation of T cells. But it is amazing that it took us such a long time. I mean, we're known for many years before that it's an inflammatory disease, and such as, for example, also diabetes, rheumatoid arthritis, and MS. And in these diseases, I think surely 20 years before us, they realized if they have inflammation, there must be an involvement of the immune system and adaptive immunity, and started to study that 20 years before we did that in cardiovascular disease, which is amazing that it took us such a long time to realize this.
So the autoimmune component of those diseases?
Yes, there are. Yeah. But if you have inflammation is always regulated by immunity. And we started to look at inflammation as something isolated.
Right.
Caused perhaps by oxidized LDL and had inflammation, not realizing that the immune system regulates inflammation, and you can't study inflammation if you don't study
the immune system.
Exactly. They go hand in hand.
We're going backward.
Yeah.
And also when we started this 92 or so,
right.
The idea we had was exactly the opposite.
That it would accelerate atherosclerosis.
Yes.
You thought there was exactly.
We thought this was an autoimmune disease. That's why it took us a long time to publish it, because we were out to prove it was an autoimmune disease, and we got exactly the opposite.
Because you were looking for immune complex?
We imagine that immunizing with oxidized LDL will actually accelerate.
Just like kidney disease and
Yeah, it
others.
will accelerate. And when the reverse came true, we didn't believe it first.
So we did the study once more.
We did it twice. Exciting, this is exciting.
And the funny thing is that Palinsky and Whitson thought the same thing.
Same thing.
They were also out to demonstrate that this was an autoimmune disease where this autoimmune response caused inflammation. And they also redid the study.
Yeah. They used a lot of it.
This is awesome.
Yeah.
It's exciting.
So you're overall thinking about how an agent of disease such as LDL becoming oxidized is thought to be an agent, and now suddenly can be turned around as a
Protection.
yeah.
Or the response to it. Right.
So this whole discussion is sort of an introduction, a conceptual introduction. I have to follow along, try to understand what you're thinking. But just to be able to follow Jan's discussion, I'd like to know, what is your overall thinking about how that happens? Why does, if oxidized LDL is a part of, as an agent of what is going on in the cell wall,
Right.
then injecting oxidized LDL nevertheless accomplishes the opposite. And what is your, how do you think about this? What is your concept?
I think I can confuse
Yeah.
because I think that's coming back to the balance between what's done by the innate immune system and what's done by the adaptive immune system, which has balancing effect. And this, you all know this. Well, that is an infiltration and accumulation of LDL in the matrix. It becomes aggregated, oxidatively modified by some mechanism that we're not sure, but we know that will happen. It's then recognized by, and this induces inflammation, and we have an infiltration of the monocytes which turn into macrophages. And these macrophages express a number of scavenger receptors and toll-like receptors that is part of the innate immune response. And they can react with a lot of what's called PAMs, pathogenic molecular patterns, which are present on apoptotic cells, on oxidized LDL, and on certain number of bacteria. And it's a sort of rapid but rather blunt defense system. But it's not very specific. And I think that's a beautiful study by PK's group, that if you block the toll-like receptor, toll-like receptor 4, you can reduce atherosclerosis to about 50%.
25% by TLR, and then there's a downstream adapter molecule from TLR called MIDI88, and knocking that out, 50% reduction.
That's just like paralyzing macrophages.
Yeah.
Well,
Just published in VNA.
that's like a fundamental chemotherapy.
That's
against
why we
immune
published in PNAS, because it was a fundamental.
No, it's a fundamental. I mean, you do chemotherapy against immune
Right.
system, and that probably unlikely to be a therapeutic pathway.
No. You don't start thinking remedially. It's right.
I know.
Understanding the concept first.
Yeah, I understand.
Because, of course, if you completely block innate immune system, we may not live.
You said something
But
right.
then there are specific ways of influencing pathways that leave the general protection
Outside.
intact. And still,
What it shows very nicely is that the innate immune response to hyperlipidemia is
proatherogenic.
Is
Atherogenic.
atherogenic.
The innate immune response
The innate immune response against
drives disease.
this is innate, right?
Yes, it's innate.
This is right here.
Now is the innate
You-
immune receptor.
You are at the speed of light here, because there are a lot of things happening here. Do you believe that our oxidation process is happening right sub-endothelial? Is that what you're thinking?
I think that's one place that it can take place.
Do you, no, what is your feeling? The reason I'm saying this, when we just went around and
To answer your question, we did a study where we injected LDL, and then it gets deposited in the rabbit vessel wall, and it gets oxidized there, and the oxidation epitopes appear first in the vessel wall, not in the circulating blood.
not in circulation?
Not in that particular model. This was done by Adam's group.
And it only happened in those high shear stress, low shear stress area, or the entire vessel wall had Or you had this, if you put them on the fluorescence, you see the strips all over?
In this model, it wasn't the entire aorta, but it was an artificial model. We injected human LDL into a rat. And then these LDL particles accumulated in the rat aorta, the entire,
The entire aorta.
after 12 hours, and you could see oxidized epitopes after 24.
I have a feeling if you picked up other arteries or even some other tissue, you might have found some of those oxidized LDL. And my feeling is, it's not just the shear stress or the pathology here, it's the mitochondrial burst and creating all those free radicals, and they affect everything.
Actually, maybe even more than that. Once LDL goes in, it interacts with proteoglycan.
Proteoglycans, right.
And that interaction is critical for it to be retained.
Right.
Because if you modify interaction between LDL and proteoglycan so that it doesn't interact, you get drastic reduction in atherosclerosis, despite cholesterol levels of 2,000.
You mean the residence time basically is modulated.
Besides entry, there has to be interaction with proteoglycans. If you create LDL that is defective in that interaction, you can raise the circulating cholesterol to 2,000,
Right.
get about 80% less atherosclerosis.
Because they can't go to
Interact, correct, it is not retained. So retention is a very important prelude to modification.
So that's you're calling it retention, the old-fashioned way is residence time, correct? That's what
Response
you're,
retention,
if the residence time is reduced, it's like less traffic jam there, they can go
You
fast.
may not get atherosclerosis even though your cholesterol levels are 2,000.
Well, my question was, where does this I have a feeling that modified LDL and oxidized LDL is happening outside of vessel wall,
may.
in the circulation, simply just because you have so many oxidative agents attacking molecules left and right.
Circulating blood has a huge antioxidant defense system. That's
Okay.
why we don't think
Probably liver. If you pass through liver where the
happening in other organs.
liver is a fireplace, this cytochrome 450, and all that, things that are around there. That's the war zone. If they pass through one time, might be a good idea just to take a little blood before liver, right after liver, and see oxidized LDL has changed, or other, organ. Because it's very unlikely that you only have. Nawab and his group thinks that oxidized LDL is produced right here.
Well, I think there's a lot of evidence to support that, but it may be occurring in other organs.
But we haven't studied other organs.
It may be happening in other organs.
In your picture, you had oxidized LDL in the blood.
Yeah.
How much of the LDL in the blood is oxidized? And
Wolfgang has actually done the biggest study on that. But
Well, I could present those.
Yeah, we wanna-
I'll probably say a little bit later on.
Yeah, we're going to see it.
So I think that we've done some calculation on that, but perhaps about 1%.
1%.
It's at the end of.
You would assume oxidized LDL is the pro-atherogenic particle. So you would assume there should be an correlation between oxidized LDL in the blood and as a risk factor for the liver disease.
Some of the studies do show that. Some studies show it. And the idea is
You would assume
that oxidized LDL in the blood may be also coming from the vessel wall.
Yeah, okay.
But it doesn't necessarily prove that it's being oxidized in the blood. It may be leaking out of the vessel wall into circulating blood.
And if it comes in a second time, it's much more likely to become retained.
Right.
If you combine modification.
If it's already modified.
Yeah.
Yeah, I wonder if there's a piece missing from the diagram. I wonder if the clue might be in the oncology literature. And maybe it's just missed just because it's drawn this way, but we're talking about modified macrophages trafficking in both directions. The blood side, the luminal side of the endothelium is like fly tip, it's covered in biological stuff. But the medial side is like a wax spore. In the diagram, there's nothing. And surely there has to be a biology on the medial side of the endothelial cell that's similar to the biology on the luminal side. Otherwise, how do the cells find their way out? And the reason I say oncology is the same processes could apply to metastatic cells.
You're quite right in that.
Yeah.
It's been very few studies looking on that. But it's, I think also we think traditionally the concept that macrophages migrate out to me. I think that is that migrate is probably the dendritic cells, which are very, very few.
Very few.
Very few.
See, the study that Histed, the guy, the atherosclerotic ATVB guy, did with shear stress, where wherever you change the shear stress, the alignment of these cells change. And it's actually a beautiful picture. You see, cobblestone type of structure on left side, and the change shear stress is such a weird anatomy of cells. Probably that's involved. But I just wanted to get your feeling of, you think that oxidized LDL factory is here or it's outside, but the residence time somehow is affected by modification of XCL.
But we know extremely little about resident in other tissues. And I'm sure there's going to be retention of LDL also in other tissues.
In other tissues.
So there is a potential that the oxidation of LDL occurs in many different tissues?
Could be, could very well. We know for sure it occurs in the vessel wall, but it may be occurring
I can promise you, we had more oxidized LDL after we ran than we started this morning. This is just, inevitable.
But actually, Sam Parthasarathy has suggested that exercise protects you by oxidizing LDL so it can be cleared rapidly.
Are you serious?
He's actually published a paper on that.
But it's interesting, you stories are focused to sit and look on slides like this. So we don't think of anything else for the rest of the day.
Great, let's go. Wow, this is getting exciting.
So this is, of course, what it looks like. And interestingly, we know this was known for centuries that looked like this, and still taking the step toward we should know from the school books. If a macrophage take up a lot of things, you're going to present it on class 2 receptors for T cells. But it's amazingly that it took us such a long time to come up with that idea.
We are dense.
We know that macrophages eat them, but we always look at them as like a garbage eater.
Yes.
We never thought that they would break it down in polypeptide and express them, present them to T cells.
All of us read that.
Isn't that interesting? Yeah.
Yes, it took such a long time to do that. I think
So it has been confusing. We call them scavenger cells.
Right.
we just
Because we were obsessed with Brown and Goldstein on how to eat these cells. We never thought that what happens after. Actually, there were a lot of paper on hydolysing, the lysosomes, what happens in lysosomes, but it was like looking at them as like a garbage.
yes, it broke down and then disappeared.
See, you now opening a completely different view, I think it's something that we learn from this meeting, the way they talk to us, is we have to talk the same way to people out. Now I'm serious. But what is the missing point? What was the missing point that is so clear? It's right here. After they eat, their job is not just eating. Their job is to report to FBI. And their body's FBI system is right sitting there.
That's exactly. It turns out innate and adaptive immune system, although separate, they are ultimately linked.
They have to report to George Bush, though he doesn't understand.
Exactly. They are linked.
For antigen presentation, is it important which way the oxidized LDL is coming into the macrophage? The scavenger receptors or toll-like receptors. Is it important?
It doesn't come in by toll-like receptors, but it may be if it comes in by scavenger receptors, and it may be if different types of scavenger receptors all coming up. And then there is the alternative approach, which hasn't been looked at all, and that's that the immune complex is taken up.
The complement.
Well, yes, even the complement. It can come in with complements, and together with IgM. But you can also, we know we have a lot of IgG in the blood. We have the oxidized LDL, and we have the oxidized LDL. So we've got
The
a
bond?
complex. They're going to be taken up by FC receptors.
Okay.
And that's quite another pathway.
And the response of the macrophage is
Differs a
different.
lot.
Okay.
FC receptor only macrophage FC receptor?
No.
Oh,
So
T
not
cell?
other cells. B cells, for example,
B cell?
can activate.
So they can completely bypass the innate system.
Yes.
Interesting.
What is the
See, oxidized LDL does not go through the TLR receptor. It doesn't. It is scavenger. It triggers.
Oh,
It's a ligand.
I see. So it's like whatever.
TLR is the receptor. Oxidized LDL is the ligand. It doesn't go.
It doesn't scavenger. Okay.
It's
Well, TLR is not a scavenger either. It's
a signaling.
It's a signaling pathway. I see.
It's toll-like receptor, a signaling receptor.
signaling.
It does not take up.
No, it doesn't take up, it's a signaling.
Scavenger receptor which takes up but doesn't signal.
But don't you think that the initial, the minimum amount, where it's a dose-dependent probably, is more true as scavenger. So you have, if it is a small amount, scavengers are going to take them up and clean. But if it's too much, probably they go saturate the scavenger receptor and they affect TLR. Is that
Actually, the scavenger receptor classically is not a saturable pathway.
I mean, they keep eating.
It doesn't downregulate.
Not
Unlike the classic LDL receptor,
No,
that's
no.
the difference.
okay.
It's a non-downregulatable.
Not downregulate. Well, I mean, it's traffic. You have a lot of these particles here. While they're waiting, they can trigger TLR or others. Whereas if it is just a few, they're going to clean up. And
I don't
we don't know.
I don't think we know that, all that detail.
So this link came when?
This one?
This click came in late 80s?
Yeah, 1988. But it was so we were so slow. We were amazingly stupid. We weren't sure if there are activated T cells.
We're not going to write this in scientific American. We're going to have to say gap here. Actually, it's good to show a gap here. This is great.
But it was, I think, in the late 80s, we realized that there is an interaction. There are T cells there must be an antigen presentation, so there can be an adaptive immune response.
When was the first dendritic cells was found? Do you know? When the first, because T cell was like 84, 5.
In the atherosclerotic.
Atherosclerotic plaque.
I think only in
Which was like last 4 or 5
years. That Bobrskov from the Russian, he's the one. He's the one who identified
dendritic cells.
Well, what was the relationship between macrophages, dendritic cells, and the foam cell?
Almost all the same, right?
But they can be, but they have different functions.
Macrophages, dendritic cells, and also some B cells are antigen-presenting cells. They can activate adaptive immune response. The dendritic cells is the most specialized and the most powerful.
In terms of antigen presentation.
And it's all over the place, and it migrates around all the time. So it
And a macrophage can turn into a dendritic cell.
Yeah.
It's the same family.
And the foam cell is that?
Foam cell is lipid-laden macrophage.
And it's not necessarily a dendritic cell that's gone foam cell.
No.
Dendritic cells, from physics point of view, has most exposure area than any other cells because they're
Got these pseudopods.
Yeah, pseudopods. So they have more area to click. Because everything here is happening by chance. We know that there's no eyes here to go after. So it gives a high likelihood of clicking to the antigen receptors.
So macrophages are not necessarily macrophages. There are species of them that then generate or become dendritic cells.
Exactly.
Species of them that become.
In the atherosclerotic medium, you can identify macrophages that have no lipid and macrophages that have lipid.
Yeah.
I mean, that's been known.
Right.
So why does it become immunogenic? It's an endogenous particle. We should be tolerant against it. We shouldn't recognize ApoB or oxidized LDL because an endogenous
Part of our body?
Yeah, part of body. It should have tolerance. But there is such dramatic changes in oxidized LDL during the oxidation. So it produces a number of epitopes that are not recognized as self anymore. And the most important thing is the fragmentation of the protein part, ApoB. Then you have the oxidation of the phospholipids that PK talked about. Perhaps most importantly, when the fatty acids are oxidized in the phospholipids, they generate a lot of very reactive compounds, among them aldehydes. And the aldehydes form covalent links with amino acids in the breakdown products of ApoB. And this is called haptenization, and this is the most powerful way to make an endogenous peptide antigenic.
Antigen.
Yeah.
Changing this cova.
Yeah.
Malondehyde, whatever?
Yeah, malondialdehyde and 4-hydroxynonenal.
I see.
So if you if you break down the protein, you're going to change the three-dimensional structure, and then you add an aldehyde. Then you really changed something there.
And this is like a red flag for our immune cells. That they just
Well, that's what we call a neoepitope.
neoepitope.
New antigens are exposed. created.
Now, but you said it is endogenous. Do we know that LDL or oxidized LDL go through umbilical artery to the fetus?
Well, you can demonstrate cholesterol deposits in the arteries of aborted fetuses of hypercholesterolemic mothers in animals and humans. So the answer is yes.
Well, that can be gene. They can have the same gene in
No. If the mother is hypercholesterolemic,
Yeah, but if it is a fetus, if it is a familial,
the fetus doesn't synthesize cholesterol at that early stage. It's all dependent on maternal.
oh really?
Yeah, so aborted fetus of a hypercholesterolemic mother will show in the aorta already lipid accumulation.
That is so interesting.
It's been in this group,
Kalinske, no.
Alinsky's group, yeah.
But,
There's an Italian.
Napoli.
Claudia Napoli. Yeah.
And if you look on newborns, they have fatty streaks when they are born.
Well, newborns, yeah. I can say because they have liver and they have. Are we sure the body at that stage cannot create
It's
Minimum. Then if you look 1 year later, no fatty streaks.
It regressed after birth.
Wow.
And then it comes back again, worry about that.
It starts again at 8 years
So in the very early stage, the fetus, because the cholesterol is determined by
By
the
nose.
mother.
By nose. So, and that's a huge cholesterol. And then
Goes down.
goes down. Oh, didn't know that. There is something right here to learn, right?
Cholesterol is also low in newborn and there too.
Wow.
Yeah, they showed that in the LDL receptor knockout and human fetuses, both.
When asked about this, the fragmentation about this is evidently very important.
Yeah.
What is that? I mean, is that a proteolytic process, and is that lysosomal, or what is it?
No, it's not lysosomal. It occurs in the extracellular matrix. I'm not sure if they identified the exact enzymes that do this. But you can show very clearly as the oxidation process goes on.
In the extracellular
In the extracellular matrix. So it's not lysosomal.
It's just protein degradation, just like any other unfolding of things that happen is by chance.
Something changes in the ApoB, electrophoretic mobility change.
But we know there are not too many molecules like lipoproteins in the body, right? I mean, these are huge. They're almost close to half of a cell, right? 30 to 50 nanometer each LDL particle. That's a huge, like 10 of them are going to be almost a cell, a small cell. So they're, from nanotechnology point of view, they're huge spheres. So that might be a reason they get degraded or some sort of fragmentation. Okay.
Well, and this is what it looked like. So you have the fragmentation, you have the oxidation of the phospholipids, and you have the aldehyde modification of the protein. And this is going to be recognized by different antibodies. And interestingly, if you look on the oxidized phospholipids, they are only recognized by IgM. But if you look on the protein part here, the aldehyde modified protein part, they're recognized both by IgG and IgM.
I see. And do we know what exactly is the chemical sequence that they...
That's the whole.
That's the peptide study. Okay, but that's for peptide, but not for phospholipid. Phospholipid we know that. And that is a very common,
That's in apoptotic cells.
That's in CRP.
Exactly. That's why the IgM is the innate IgM that we all have.
Okay. And do you know that this guy studied and he found 99% familiarity between the behavior of CRP and IgM in early species, and this guy is a rheumatologist, Jerry Gruson. I have his name. I invited him for a talk, and he gave a deep discussion that he believes from the phylogenetic cells, or the evolution, CRP and IgM were the same.
Yes, it is part of the innate immune system. CRP is part of the innate immune
Innate
system.
immune system.
Yeah, and I think that's an interesting balance between here, because IgM traditionally, oversimplified, is innate, and IgG is adaptive.
Adaptive.
So by looking on the balance between these two, we might get an idea of the balance between the innate
Innate and
adaptive immune
IC.
response.
And do we know what is the ratio, like 10 IgM, 1 IgG, or 1? Because we know that phospholipid is a lot more than apoprotein, the total area or total mass.
I have some good slides.
Okay.
So what we know about the antibodies is that in humans there is a mixture of IgM and IgG, but IgM appears to be more common, but it's difficult to say because IgM is not that specific, so one IgM could bind to several epitopes, and then you think that it's more of it because it binds more places. But there is more IgM, but there are both types, so we have both innate and adaptive activation. The antigens are the oxidized phospholipids and the aldehyde-modified peptide sequences. It's not one epitope, it's a large number of different epitopes, and we know that this is an endogenous response. You can see very clearly if you have the APE knockout mice, for example. If you have ordinary show, they don't have any autoantibodies against oxidized LDL. If you induce hyperlipidemia by high cholesterol diet, you get a tremendous increase in autoantibodies. So it's
And both, the IgG and IgG?
both IgM.
No,
Primarily IgM.
IgM.
Primarily IgM. This is a little summary about what we know about the adaptive response to it, which is, this is an area where it happens a lot at the moment. During the last 2 years, a lot of this has been elucidated, and we know different parts are doing. So we have the uptake, the presentation, and we have two receptors that are at least characterized by now. It's the class 2 receptor, which is the classic peptide presentation.
MHC class.
MSC class. So that's what Hansen is doing, crystallography?
Yes. Yes, he uses that one.
Yeah.
And then it's a new one that we don't know so much about. It's the CD1 receptor. And that's particularly interesting because it presents lipid-associated antigens, which should be of particular interest. So we think that the phospholipids end up here, and the peptides from the control end up here. And if we go this pathway, we have an activation by specific T cells. So it's not just any T cell. It has to be T cell that specifically recognize this epitope. And then from here, it can go in different pathways. And that, where it goes, is probably going to be very important for atherosclerosis. One pathway is called the Th1 pathway. These T cells activates the release of cytokines from macrophages, pro-inflammatory cytokines. And this pathway has been rather well characterized. This is a pro-atherogenic pathway.
This is where you have interferon gamma IL-12. So pro-atherogenic.
So if you knock any of the cytokines that are released in this pathway, you get less atherosclerosis. This is a pro-atherogenic pathway. Then another pathway has the Th2 pathway. And that's a protective pathway. It's anti-inflammatory, so it downregulates the secretion of gamma interferon and TNF, and it does so by secreting IL-4 and IL-10. So it's an anti-inflammatory pathway.
Let's have a question from a lay person who doesn't know a lot about these cytokines. There are like 50 some different type of